Today we will discuss about the DNA sequencing. It is important topic for PGT biology exam. Now what is DNA sequencing? It is a fundamental technique which allows the scientist to determine the exact order of nucleotide in a DNA molecule. किसी DNA molecule के अंदर न्यूक्लियोटाइड का एग्जैक्ट ऑर्डर क्या है उसको डिटरमाइन करने की जो टेक्निक है दैट इज कॉल्ड डीएनए सीक्वेंसिंग. नाउ हु is related to sequencing DNA sequencing work? So most importantly, Frederick Sanger. He pioneered the Sanger sequencing method in 1977, so it is also important. Now there are different types of DNA sequencing like Sanger sequencing, which were discovered by Frederick Sanger, then next generation sequencing and single molecule sequencing. Now what are the application of DNA sequencing? So it is helpful in genome sequencing. किसी organism का complete DNA sequence करना होगा, genome sequence करना होगा तो उसमें helpful होती है. Metagenomic sequencing जो कि एक्सटर्नल जो एनवायरमेंटल सैंपल्स होते हैं वहाँ से किसी जेनेटिक मटेरियल को स्टडी करने में हेल्पफुल होता है, उसको सीक्वेंस करने में, डीएनए सीक्वेंसिंग करने में हेल्पफुल होता है. देन ट्रांसक्रिप्टोमिक सीक्वेंसिंग. सो नाउ लेट अस डिस्कस अ फ्यू क्वेश्चन आंसर्स बेस्ड ऑन दिस टॉपिक. क्वेश्चन नंबर 1, हु पायोनियर्ड द Sanger sequencing method? So it is easy one, as we discussed. Frederick Sanger pioneered the Sanger sequencing method in 1977. Question number 2, which DNA sequence technique allows parallel sequencing of millions of DNA fragments? So option number C, next generation sequencing. It is helpful in sequencing high throughput sequencing by simultaneously sequencing millions of DNA fragments. Question number 3, what is the principle behind Sanger sequencings? It is an important one. So option number B, synthesis of DNA strand using chain terminating nucleotide. It is the basic principle behind the Sanger sequencing method. Next is, what is the primary application of metagenomic sequencing? So as we discussed earlier, option number C, analyzing microbial communities in environmental sample. Question number 5, what is the key advantage of next generation sequencing over the Sanger sequencing? So it provide a large amount of sequencing at lower cost. So option number B is the right answer, lower cost. Question number 6, which of the following is not a type of next generation sequencing? So there are 3 types: Illumina sequencing, ion torrent sequencing, and Oxford nanopore sequence. So option number B is right answer of this question, Sanger sequencing. Question number 7, what is the significance of single molecule sequencing technique in DNA sequencing? So option number B, they provide longer read length. Question number 8, in transcriptomic sequencing, what type of RNA is typically analyzed? Important question. So option number A, that is mRNA, messenger RNA. What is the primary goal of metagenomic sequencing? What is the primary role? Option number C, that is analyzing the genetic material of microbial communities. It is a repeated question. Question number 10, how does nanopore sequencing differ from other sequencing methods? So option number C, it directly reads individual DNA molecule.